So hi everyone, today I'm going to perform quantitative plating experiment. You could see this would be our raw milk, and if you work in a laboratory, you don't drink it. And so for performing today's experiment, I'm going to prepare two empty test tubes. One, I'm going to get the raw milk inside, exactly same raw milk inside of these two test tubes. One, I'm going to label pasteurization. Pasteurization tube going to go to water bath, 63 Celsius degree for 30 minutes. Another, I'm going to just let them stay in the room temperature for 30 minutes. So this would be two groups that we are going to perform the experiment on. So as I just talked about, there are two exactly same test tubes of raw milk. So I'll get enough milk. Since they are almost just the same test tubes, getting the same raw milk, the only difference is one of the tube, one I labeled pasteurization, pasteurization. Another I labeled unpasteurization. So this indicates I am going to let this test tube stay in the room temperature, whereas this test tube I labeled pasteurization, I'm going to put them here into the 63 Celsius water bath for 30 minutes. And this is a method we call pasteurization. So now let's count the time for 30 minutes. The purpose of performing quantitative plating. So you now know the idea of quantitative plating is a method to count the cells. And you also know there are other methods to count the cells. You could go to the slides. There are two other methods we mentioned in the slides. But for here, quantitative plating, we are going to first perform pasteurization experiment, compare these two test tubes, and then we are going to do series dilution. After series dilution, we are going to mix all the series dilution samples with molten agar. So here we prepared all the molten agar, and after mix all the cells with molten agar, we plate. So this is the reason we call plate method. And then we are going to plate all the molten agar mixed with our samples onto petri dishes. And probably after 2 day growth, we will see the result. And then we are going to count the cells. We are going to count the cells. What's the difference between the pasteurization and the unpasteurization samples? And then you should be able to get an idea why in the market you purchase some milk, they all label pasteurization, because this is a method to kill the cell. Okay, I'm going to do the plating method now. Here I prepared 3 water bottles, and one I labeled -10 to the -2, 10 to the -4, and 10 to the -6. And now they basically contain exactly the same volumes of distilled water. What I'm going to do is I'm going to have a large micropipette transfer 1 ml of my unpasteurized raw milk samples into the first water bottle, and then I mix it, shake it, and transfer 1 ml to my next bottle, 10 to the -4. Shake it well, and transfer from the diluted sample from 10 to the -4 to 10 to the -6. So this is a method we call series dilution. And since this is unpasteurized raw milk, I'm going to do exactly the same thing later for pasteurized raw milk after 30 minutes. So this is during this 30 minutes, we are performing the unpasteurized raw milk. So here As showed you in the slides, exactly the same. We have 10 to the minus 3. We are going to get 100 microliter from 10 to the minus 2. 10 to the minus 4, we get 1000 microliter from 10 to the minus 4. And then 100 from 10 to the minus 4. So I'm going to show you how I'm going to follow in the slides. I get unpasteurized raw milk. I shake it a little bit, and then I am going to get 1 ml. So after this, we get 10 to the minus 2 dilution. What we are going to do next, we are going to transfer 1 ml to this 10 to the minus 4 bottle. I shake it well, so I'm going to use a new micro pipette. Remember, I change my tip every time. Shake it. And then I'm going to transfer from 10 to the minus 4 to 10 to the minus 6. Shake it well. After that, I'm going to start from the lowest dilution, because if I start from 10 to the minus 7, I guess the lowest dilution, I could use the same tip. So in order to use the same tip, I'm going to start from the lowest dilution. 100 from 10 to the minus 6. So this is 100, and then 1000 microliter to this plate. So this is the method we call quantitative plating. Then actually I could use the same tip. So 100 to here. 1000. So I need to mix my sample with molten agar, one at a time. And in order to prevent contamination, I use a paper towel to wipe the out layer. And the method, quantitative plating method, would be like this: mix my sample with molten agar, and I swirl it to evenly distribute my sample, like this. Then I wait it to solidify. So agar would be solidified later. So now it's a timing for all the agar to solidify. After 30 minutes, we are going to do exactly the same thing to seri-dilute our pasteurized raw milk. And we are going to see the result 2 days later.